Okay, hello. Welcome to the Global COVID Lab meeting. My name is Sabrina Welsh, and I'm the Director of Programs and Operations at the Human Vaccines Project, and I'll be your moderator for today's webinar. I'm going to share my screen. Okay. I wanted to start by thanking all of you for your interest in the Global COVID Lab meeting. More than 900 people registered for the series, and we had attendees from over 30 countries join the first session. We at the Human Vaccines Project started the lab meeting to bring together the global scientific community working on COVID-19, and we're so happy that so many of you are joining this discussion. Just a quick note before we begin. The information presented today is pre-published data and is currently under peer review. At the request of the author, we ask that you not reproduce or disseminate the data presented in any way. The session will be recorded and available on our website and our social media channels for you to review. And for this particular webinar, we will delay the posting of the recording until the data is published. We are very excited for today's speaker, Dr. Bette Korber. Dr. Korber is a laboratory fellow at the Los Alamos National Laboratory in the Theoretical Biology and Biophysics group. She leads an interdisciplinary team that provides bioinformatics, theoretical, and statistical support in collaborative efforts with experimental researchers. Her team's focus is on the areas of HIV viral diversity, the human immune response to infection, and vaccine design. She has also worked on filoviruses, hepatitis C, and influenza viral evolution and vaccine designs, and, like so many, has recently begun to work on coronavirus. Today, Dr. Korber will present her preprint report that has garnered much attention over the last few weeks. Her group has developed an analysis pipeline to facilitate real-time mutation tracking in SARS-CoV-2. Dr. Korber will give us some insight into mutations on the spike protein, which may be especially relevant as spike is the target of most vaccine strategies and antibody-based therapeutics currently in development. These findings have important implications for understanding SARS-CoV-2 transmission, pathogenesis, and immune interventions, and we are very fortunate to have Dr. Korber with us today to discuss this research. During the presentation, all attendees will have the opportunity to send questions about the data presented today using the Q&A function in Zoom. I'll collect your questions and ask Dr. Korber a broad selection of these questions after she has finished her presentation. We'll have about 25 minutes for discussion, and we hope that you'll take the opportunity to ask Dr. Korber your questions about the data presented today. It is now my pleasure to introduce Dr. Korber from the Los Alamos National Laboratory.
Hi, can you hear me all right? Is this working?
Yes. Yep.
Okay, I'm just going to share my screen and my slides.
Great. Okay.
And let's see. Oh, there we go. Okay, so I'm seeing myself over some of the slide. Were you going to set that so they were separated, or is this going to work?
Yes. Yep. Right now I've got you side by side, so the recording will have it.
Okay. So this is the way I can't see all of my slides, but you can. Okay. I think this will work. So I'm going to be talking about in particular, one spike mutation that's really come up rapidly and has now changed spike globally. The earlier Wuhan form of the spike has now been pretty much replaced by this single mutation in spike. And I'm going to talk about how that happened over the course of the month of March. And also talk a little bit about recombination at the end. And as Sabrina noted, this is not published work, so this is just an update on our ongoing work and intended for discussion in a scientific audience. So I would appreciate if you didn't redistribute these slides, and it's currently under peer review. So basically, the mutation that I'm going to be talking about in spike is a mutation at position 614. And it's part of a haplotype. It's got 3 mutations that accompany it as it moves through the world. And, the genome of this virus mutates very slowly, so it doesn't acquire mutations quickly. But there are these 3 particular mutations that define this haplotype. At position 3037, there's a silent mutation. At 14408, there's a mutation that causes an amino acid change in the polymerase, and then there's also this D614G mutation in spike. And the reason I'm particularly interested in this one is because we're all working with spike to develop vaccines and to develop immunological reagents that might be used for therapy. So it's a key protein in our combating this virus. And I certainly didn't bring up this mutation in the first place. It was done by GISAID. So for those of you who don't use GISAID or aren't aware of it, what GISAID is a remarkable database, and it's a remarkable global effort. So people are sequencing this virus all over the world, and when they when they get sequences, they upload them into GISAID, and GISAID now has tens of thousands of sequences. They're coming in at an extremely rapid pace. And so everyone, you're in a fire hose of data. You're getting sequences from all over the world. They're filtered through GISAID and provided back out to the world so people can study them. And GISAID early on noticed that there were three clades that were pretty common and dominant, and they started tracking them. And so this is their tree from yesterday. And the one that we're interested in for today's discussion is this haplotype that's got these three bases. And it's the one that's marked in this kind of orangish color. And they call it the G clade because it was the D to G mutation in spike. And they have two other clades that they're tracking as well. And you can see the frequency of these things over time. So what I'm going to be talking about is this increase in frequency. And there have been several papers published about this particular mutation and set of three mutations, because it was coming up pretty early on to be common and was very common in Europe. And so there have been a couple of publications that talk about it being the dominant form in Europe. And I think a lot of the initial thought about this was that it was really a founder effect in Europe and spread throughout Europe. And I'm going to try to make a case that it's actually under positive selection because of the way it's evolved. So out of 10,160 sequences last week, when you look at these three bases that define this haplotype, you see that either having one form or the other, the orange is always going to be the original Wuhan form of the virus, and the TTG is always going to be the form that has the G spike, and it's going to be in blue throughout all of my slides. So the original is orange, and the later form is blue. Having a preserved haplotype is really common, 98.7% of the time. So you don't very often see mixed haplotypes. Occasionally you get them, and occasionally you get ambiguity codes. The first sequence that had all three of these mutations was in Zhejiang, China. And there was a couple of other sequences in that time frame that had two of the mutations, and one of those was the first case that had this D16G change in Europe, and it happened to be a German case. And so these were coming up in late January. What's happened since January is that there is many samples sequenced from all over the world put in GISAID prior to March 1st. And this form was only 9% of the sequence set prior to March 1st. During the month of March, it became 66% of the sequence set, and post-April 1st, it became 83% of the sequence set. So you can see it's now the dominant form that's being spread in the world today. So most of the spikes carry this mutation now. And the reason I believe this is an important issue to think about is that spike-based vaccine reagents and antibodies are generally based on the Wuhan reference strain. So the fact that we have this amino acid change relative to what we're using for our vaccines means that we need to know what it does. We need to know if it does impact the vaccines or not. So it's just going to be an important test reagent in our panels for when we're looking at vaccine responses to make sure that it hasn't impacted the value of the initial response. And it well might not, but it's an important thing to know. So I'm going to show you a lot of these kinds of graphs during the talk. And this one is done globally, but we can do this on any region or city or country within the GISAID database that's specified. And what you can see is the orange form, the original Wuhan form, was pretty much everything that was sampled until the end of February. And this is a weekly running count. So this is counts. And then the blue form, or the new mutant form, starts to come in. And then you have both forms present, but as you proceed through time, this form diminishes till later samples are almost entirely the blue form. So you can kind of think about this. If you go to a certain height in the bar chart, these are the number of sequences tallied that week. And you can see there's a high proportion of orange here, and the orange is almost gone here. So we're really replacing this virus. This is another kind of plot of exactly the same data. So here you see the original Wuhan form of this haplotype. And then you see the blue form coming in, and these are now daily cumulative values. And you see about March 31st, you basically have no more increase of this original Wuhan form sampled. All the increases are in the mutant form. So this is another way of viewing this, and we have developed a site called cove.lanol.gov, and you can recreate these maps, and you can recreate a lot of the figures I'm showing you at this site. And this is really a pipeline that was developed by my colleague Will Fisher. And what he does is he takes the GISA data, and we upload it every day, and maybe sometimes not quite every day because it's quite a bit of work. And then he filters out the best sequences, the ones that are the most intact and have few ambiguous spaces, that sort of thing. He aligns them all, and from these alignments, we make trees. There are other sites that are making beautiful trees, but we also follow global tracking of really any mutation you want to look at. In Spike right now, we're going to extend that to the full proteome as we go. We haven't gotten that far yet. But here you can see one of our global maps tracking just this mutation. So this is through March 1st. These are the samples that were available. And you can see that it was coming up strongly in Europe, and people were very aware of this. It wasn't present very much in the United States, and it was barely present at all in Asia. And by mid-March, or the end of March 21st to 30th, so just 1 month later, all over the world, it had really begun to take up the samples. And the size of a circle in these cases indicates the relative frequency of samples within one of these global charts. So the United States is going from predominantly a original form, Wuhan form epidemic, to a predominantly G-clade. And then this is now April 21st through 30th, and you see that it's even more blue in the different regions that you look in. So what we're going to do now is go sort of region by region and break this down, getting more and more local in Europe and in the US and in Asia, and see the transitions in time. And the reason I think we've got support for something other than a founder effect is that every single place you look, when this thing comes in, it starts to take over that local epidemic. So the pattern repeats, and it's the repeated nature of the pattern that I found really striking. So it's recapitulated at every geographic level for which there's enough data to look and enough time to look. You have to have a few weeks of data, and you have to have more than just a couple of samples. But if you look at the continent levels, if you look at the country level, if you look at states and other major country subdivisions, if you look at counties and cities, however you look, when this thing comes in, it begins to dominate the epidemic sampling over the next few weeks. So again, it's the repeated nature that's really interesting. Significant shifts are observed within just a few weeks, which was really striking to me. The increase in G614 frequency is often initiated after national or state lockdowns, and so serial reintroduction of the mutant form seems unlikely. And the other thing is it's often coming into a very well-established epidemic, so the whole region seems to be carrying the original Wuhan form. This one comes in, and then there's a shift. So this is the pattern that we're seeing. So I noted that we have to have enough data and enough time, and I was sort of doing this hand by hand, country by country, region by region, and seeing this, but I thought a better way to portray it would be to have it all brought into one page. So we needed some criteria of what was enough. And so this is what we wound up defining. Onset is the first day for which there's at least a mixture. So at least you have 15 sequences that have been sampled by this date, and both forms are present. And so you have at least 2 of the least frequent form. You have data that is available at least 2 weeks later. So it's not just a very short period of sampling. And then you consider all of the sequences that were sampled up to the time when you begin to have this mixture, and then all sequences sampled after the 2-week delay date, and you require at least 15 sequences after the delay date. And then just count the Ds and Gs before the onset and after the delay, and test whether the mixtures of Ds and Gs change significantly using a 2-sided Fisher's test, and then do bar graphs to show how the fractions changed between onset and delay. And this way we were able to get all of the data from GISA that met these criteria into one plot. And what you can see is the following. If we look at the continent level, This is when the mixtures first were apparent on the left, and you can see it's usually the Wuhan original form. And this is 2 weeks later and beyond. And this is the frequency now of the mutant form, the blue bar. And you can see on every continent, Asia, North America, Oceania, Europe, South America, you can see the shift going down. And the reason Africa is here is not here is because there just weren't enough sequences yet from Africa to really look at this question. You can look at this then at the country level. And so every single country that you see a shift, the shift always goes down except for Iceland. And I'm going to talk about Iceland more in a moment. But you see, if you start in very well established epidemics that are basically the original form, you start to see the shift. As soon as this thing comes into that population, within 2 weeks later, you begin to see these shifts, and they're highly significant. And again, Iceland is the only place where you saw a significant shift in the other direction. If you get even more regional and you start looking at local regions and cities, and this is often available in GIS-8, not always, you can see that this is happening in states and in counties. So here's Washington State, here's King County in Washington, and here's California. So you can see there are cities in here, and there are larger regions, but these are all smaller than a country. And it happens whether you start with a predominantly Wuhan epidemic or whether at the time of sampling, you're starting with a predominantly mutant form. In either case, you get the shift in frequency within about 2 weeks, and again, except Iceland. And these two are exactly the same data sets, actually, because everything from Iceland was from the same city. So this shows up exactly the same twice. So now we're going to start taking these continent by continent and breaking them down. So over here on the left, you can see Europe before March 1st. And you can see that there was quite a bit of the mutant form in Europe, but it was not everywhere, and it wasn't the dominant form everywhere. By March 21st to 30th, it becomes the dominant form in every country in Europe. And here's one of those plots that show the weekly running count. And these purple lines here are the close down days for the different countries where people were asked to stay home. And so at this point, you would be unlikely to have many subsequent introductions. So let's just look at a couple of these. So you can see the United Kingdom had closed down days in late March. There was quite a bit of the original Wuhan form in this epidemic, but by 2 weeks later, it was really diminishing, and now it's almost all the new form. You can go to Spain and you can see a similar kind of epidemic where the Wuhan form was very well established in the early sampling dates. This thing came in and it's replacing it. Germany, the Wuhan form was well established, and then it changes. And then in France and Italy, almost from the first time of sampling in March, it was already the mutant form. So these are the very dark blue bands or circles to begin with here. But even in these countries, in the very early dates, the couple of samples that came in were the Wuhan forms. So sometimes we've already got the shift happening prior to the beginning of sampling. And a lot of people were thinking that this was seeding the rest of the world and making a founder virus. And I think the argument against that is just the consistency of the pattern. So now we're going to look at England broken down into Wales, Scotland, and England. And you can see the same thing happening. You can see a very, very well-established Wuhan-like form of this haplotype in March. And then when this thing begins to take over, it really soon dominates the epidemic. And I actually can't see these slides. They're blocked in my view, but I hope you can see them. And you can see this loss is often happening well after the shutdown date. So it's happening when the region has been closed. And if you look even tighter, and you go now to two very local regions in England. So now we're going to take this and we're going to take subsets. We have 1,000 data points from Sheffield because we're collaborating with a wonderful group from Sheffield. And you can see in this particular city, we had a really well-established Wuhan epidemic, and then we began to lose it, and finally it's not being sampled at all anymore, and this is their shutdown date. And I'm not quite sure the exact shutdown date in Yorkshire, but the same pattern. So this is happening at the continent level, it's happening at the country level, it's happening at sub-country levels, and even local towns. And this is really recurrent through the world, and you never see the other pattern happening where you have a mixture and the orange takes over. It's a mixture and the blue takes over in every single place we can look at except Iceland. So why is Iceland different? I think the answer comes from a really beautiful study that was done by the group in Iceland. And their period of sampling here from March 1st to the beginning of April was conducted for this study. And they published it in New England Journal of Medicine, and it's just a gorgeous paper. But this is the one place where there was a difference. So here was step.
And I'll just start from there.
Okay, super.
Okay, so why is Iceland different? This beautiful paper from Iceland explains it, I think. And if we go now to the next slide, slide 13, you'll see 4 different graphics, and it's showing how they sampled in Iceland. And what they decided to do was they sampled in the first part of March everybody that was high risk. And so that was 65% of those high-risk people they sampled because they had traveled in what they felt was a high-risk country, and mostly that was Austria and Italy. And Austria and Italy, by early March, was almost entirely the original Wuhan form of the virus, of that haplotype. And then on March 13th, they began to sample within the country a population screening where they had 85 sequences that came from people just coming, Icelanders, from an open invitation. And it's right at that moment that you begin to see the accumulation of the original Wuhan form. And then throughout the rest of the time period until the beginning of April, they were sampling a mixed population, but only 85 sequences were coming from the people who were just in the open invitation group in that middle part of March. And then the high-risk group were continuing to be sampled throughout the time period. So this is probably why Iceland is different than all of the rest of the world when we look at these statistics. If we go now to North America, we can see the same kind of pattern that we saw in Europe. So this would be the next slide, 14, entitled North America. So you can see before March, everything was Wuhan-like, and between March 21st and 30th, there was much more of the new form, the new haplotype. And it's again coming into very well established epidemics sometimes. So this is the US overall in the top left graphic, and you can see the switch going from predominantly Wuhan form through the first week in March to this new variant in the end of April, pretty much entirely in Canada, shows the same pattern. And then if you start breaking this down state by state, city by city, you can see the same pattern emerging. And again, the bright purplish magenta line indicates that the shutdown days where people started staying home. So if you look in that kind of pinkish box, you can see Wisconsin, and then you can see Milwaukee, which is the best sampled place in Wisconsin. And for example, in Milwaukee, there was a very well-established original form epidemic, and by April, late April, it's now switching in their sampling, and this is true overall when you look across the state. New York was pretty much the European form from the beginning, with just a little smattering of the original form, and it dissipated, and it's now not being sampled at all. If you look at Washington State, they've been doing an incredible job of sequencing in Washington State, and they've just got many sequences in GIS8, and you can see that in Washington State, they had this is a green box on the top right, they had a very well-established original epidemic with hundreds of samples coming in, all of the original form. By mid-March, it's transitioning, and now it's predominantly the new form, and this happens when you look at a county level as well as when you look at the state level. So the 2 counties that are best sampled in Washington are King County and Snohomish County. And you can see the transition happening in both of those counties. And again, it's happening after the shutdown date. So this isn't new introductions, very unlikely to be new introductions causing the change. And then there's Utah and Virginia, both of which started out initially having samples that, not too many samples, but the samples were predominantly the original form, and the transition happened in those 2 states as well. So there is a very consistent pattern, and I'm not showing you every single sample from every single state, but I'm showing you some representative ones, and we don't really see exceptions except one other exception, and it's California. It's following the general statistical pattern, but there's sort of an unevenness. So if you go to why California is different, slide 15, what you see there is this kind of a funny real bumpiness in pattern, and if you go between the weeks of April 12th to April 19th, that you see that it's all entirely the original form. And if you look overall, you went from all original form to sort of blue bit showing its head. And you can see there are local sampling biases that are really driving these patterns in California. And in particular, Santa Clara County, it has a lot of samples. And Santa Clara County never really had the new form become established. I think there were 2 cases of it. You see in the weekly running counts, those 2 little blue bits were exactly 2 cases, I believe. So that kind of go over the weekly running sample. And then it really never took hold. And they sample Santa Clara a lot, but they sample it unevenly in time. And so when it's sampled a lot, and San Francisco and San Diego are sampled less, then you get much higher overall rates of the Wuhan form in the overall California picture. But both San Diego and San Francisco are following the general trend. And so sort of what shows up in the California view depends on which region is sampled most over a particular week. So Iceland and California were really the 2 biggest exceptions in the entire UNTITLED database that I could find. So if we look now at Asia, go to the next slide, slide 16, you can see that Asia was obviously very well established as the original Wuhan form in the early days of the epidemic through the beginning of March. And so before March 1st, you see all of those massively orange circles, and you can see them over time for all of Asia at the top on the right, and then Hong Kong, China, Japan, and Taiwan I picked out as example countries. There's not a lot of recent sampling from Asia. So I just worked with what I had, and I had to go a 10-day earlier period than I did for the other two continents I showed you, Europe and North America, because there just were hardly any samples from Asia towards the end of March. That had really diminished by that point. But you can see that even in Asia, where you have incredibly well-established early epidemic forms, the switch is happening, and it's happening within that same few-week period. Once the blue form is introduced, it begins to really overtake the epidemic, and you kind of get this blue wave in the plots on the right, and it's happening at every region that we can look at. So on the next slide, slide number 17, my colleague Nick Hengartner thought of a really nice strategy for modeling these different countries. And so he wanted to use the same kind of philosophy that we used and pick out every single region in GISAID that had enough data to look at this. But what he wanted to do is he wanted to try to model the probability that these 2 forms were equally likely to propagate in different regions of the world. And so he constructed a time series with the number of observed D16 forms and the number of G16 forms that were sampled on every day that was sampled. And he models the number of the new G614 variant given the total number of tests as an independent binomial random variable. And so there's this probability of being G that's measured, and that depends on time. And N is just the number of strains tested on a given day. So if we look over to his black and white plot there on the right, what you're seeing is each of those little dots represents the number of samples that was available on a day, and the probability for G within that sample is indicated. So if it's down at 0, there was no G in the sample, and if it's up at 1, it was all G. And then if it's some proportion in between, you see that reflected on the graph. That little dotted line at around 0.4 is just the average for the whole sample, and the proportion for the whole sample. And so what you see is over time, you're getting this increase in the relative probability of having G, and we've seen this in the other kinds of plots as well. So the null hypothesis is that the variants are randomly observed. And he uses a maximum likelihood to fit an isotonic regression, assuming that TT is monotone non-decreasing. and does a one-sided alternative test, but we also switch this around and did a one-sided the other way to see if we ever see the original form increasing. And I'll talk about that briefly on the next slide. His test statistic is the deviance, and he does a permutation test to compute a p-value, and he does 4,000 randomizations of the data to see how likely the distributions we observed are to have occurred by chance alone. And in the next slide, I'm going to show you this breakdown by country. What he did to figure out which countries to look at is he made a minimum data requirement like we did for the Fisher's exact test that I showed you earlier. So any country that had a mixture of forms, so it wasn't just one form to begin with, like Santa Clara maintained just one form, but if there was at least 10 Gs and at least 10 Ds overall in the full sample set, and there were at least 14 testing days in the sample set, there's enough time and enough of a mixture to see if there's any changes happening. So every single data set that's a country in GIS8 he took, and every single local region he also took. I'm not showing you the slide for that just in interest of time, but essentially we saw the same kind of thing I'm showing you here. So what happens is that the probability that these are not just moving forward equally is extremely low. For many of these cases, as low as you can get, given that he did 4,000 randomizations, and all the countries where that happened are listed with the blue p-values on the right. There were a couple that were not significant, and I'm going to show those in the next slide: Austria and Greece and Iceland, which we've already covered and know about. We expected not to be. He then did the reverse of the statistic and looked to see if we ever saw a country where the original form increased relative to the other form once there was a mixture. And the only place we see that is in Iceland. So it's basically a different kind of statistic addressing the same question. And what I'm going to show you on the next slide are the Austria and Greece, the two where we didn't get a significant increase. And I'm going to show you two where we did, just so you can look at his kinds of plots relative to the kinds of plots we've been looking at. That would be slide 19. So on the left, we have two examples that are well behaved. We've got the United Kingdom and we've got USA, and you can see this step function is going up and is highly significant. And this is the same data plotted in these two different ways, the sort of blue wave plot versus the proportion of G plot. And then on the right, you can see the two exceptions where he didn't get statistical significance one way or the other, Austria and Greece. In both of those cases, at the moment we started sampling, it was already predominantly the new form, and there was a smattering low level of the other form that's just sort of maintained as a low level. So these were predominantly the new form from the very beginning moment of sampling, and just have this little bit of orange, and we don't see a change one way or the other, although Austria may be losing orange samples at this point. We may not be getting any more of those. At any rate, neither of these two were statistically significant. So those are the only two exceptions to seeing a significant change. And when he looked at the same thing on the county, city, sub-country level, he got the same thing, the same kinds of patterns. So now I'm going to talk a little bit about the data that we had from Sheffield. And this was just a lovely collaboration. We really wanted some kind of data to test this, to see if we actually could see an increase in the levels of infectivity or something like that. And it turns out those tests have been ongoing and are quite difficult to do because people are still learning the ropes with these viruses. And so being able to compare a mutant strain to the original form and have all of the proteins and the pseudoviruses and the things that one needs to do that is taking some time because they're figuring out how to do this. So David Montefiore and many others are working very hard on being able to test this, but we haven't quite gotten to that point yet. But we could look at clinical data, we thought. So I wrote to a few people at GIS8, and people didn't respond. And finally, I got a response from this team in Sheffield, and they had now 1,000 patients to look at, where they can actually link the clinical data to the sequence data. So I'm just reminding you of what the epidemic looked like in Sheffield regarding these two haplotypes. So pink again is the shutdown day for Sheffield. And they had predominantly the original orange form in the early days of the epidemic. And then that shifts, and now there's hardly any of that left, and they're getting almost all the new form. And so it was a place where there was a good mixture for their 1,000 patients over time of both forms. And the kinds of data that they had available where the people were coming in and getting tested. When they were tested, their tests went for sequencing, and they knew about these people, how many PCR rounds it took to detect a positive, and that's a surrogate for viral load. They knew whether they were admitted to the hospital, to the ICU, or whether they were an outpatient and got to go home. So we had a couple of interesting measures. We had a viral load measure, and we had a measure of disease severity, and we had the D&G data for those specific 1,000 patients. And so when I first put the archive paper out, when we were working with them, we only had about 450, but now we have 1,000. It turns, so if you go to the next slide, slide 21, what we found was that the G614 form was associated with a higher viral load. And there's a lot of overlap between the distributions, but the lower the PCR cycle threshold, the easier it is to detect and the higher the viral load. So low values are associated with higher viral load. And they changed their method of doing this partway through the study. So the last data points coming in were done with method 2, which took a few more PCR cycle rounds. And so we did a model that enabled us to use both data sets. And what you can see is there's a shift downward if you have the G61 form. So that means there's a lot of overlap, but there's a tendency to have kind of lower values if you have the mutant form. And this was not highly, this is borderline statistically significant, 0.037. When we had first looked at the data, we had a little bit higher level of significance in the Wilcoxon. We did not find the sex of the donor or the age was relevant to the PCR method, just the PCR value, so just the method and the DG status. So that was an indication that it is associated with higher viral load. And then another group in Washington that's led by Trevor Bedford looked at their patients, and they have very many samples from Washington. They can also look at this, and they got a much lower p-value than we did, but using the same measure, they were able to reproduce what we had found in our initial archive paper. So it does seem to be associated with higher viral load. What we did not find is any association with disease severity as measured by outpatient hospitalization or ICU. And in fact, the group in Washington also did not see any association with disease severity. So that's a relief. If there is any kind of association, it's going to be subtle. And we wouldn't be able to see it with the level of patients we had to look at. Okay, so now the next slide, why does this mutation possibly make a difference? We don't know. But could be virological, and so that's our first hypothesis. It turns out that this particular position is located on the surface of the spike protein proteomer, and it forms contacts with a neighboring proteomer. And my colleague Nana Karen made this slide. He's a protein molecular dynamics expert. And the thing that initially struck him, if you look at the far right on his picture, on his graphics, the top right one, you'll see that there's hydrogen bonds in the D61 form with amino acids on the other proteomer, and spike is a trimer. So he thinks that this bonding would actually potentially make the trimer more stable and have the interactions be stronger. So that's one potential difference in the protein. And if you look at the left in his diagram, you'll see the little association between S2 and S1 that he's talking about in this hydrogen bond. And so this would be lost in the new form. And so spike might be a little bit looser, more open. It turns out there's a glycan, a glycosylation site that's 2 residues down from this shift to D. And D is a negatively charged amino acid. And from our studies in HIV, we think that this might really impact the way the glycan moves on the surface of the protein, and its interaction with the protein surface, maybe masking or opening epitopes, that kind of thing. So that's a possibility, and it also can impact carbohydrate processing. when you have a charged residue that changes so close to a glycosylation state. There's also possibilities that it might modify the up-down ratios in binding to ACE2 and exposure of the cleavage site. So all of those things are possibilities, and Nana raises them on this slide and shows you why he thinks this might be the case. So there's two ways we're currently exploring this. First, by MD simulations, and those take some weeks of high-performance computing and probably won't be completed for a week or two. So those are ways to theoretically model the protein behavior and see if we can predict a difference. But meanwhile, people are looking at this in terms of structure and actually making the structure of both forms, and they're trying to look at this experimentally on the lab bench to see if any of this pans out and is validated. So that's one way this might be impacting things. Another way might be immunologically. So if we go to slide 23, one of the things that really is striking about this particular mutation is from a paper that my colleague Celia LaBranche pointed out. And in this paper, this is a paper that studied the original SARS-CoV, and it turns out that there's a short peptide that is centered on 614. So it's about 9 amino acids, and this amino acid's right in the middle of it. That's got a very high level of serological reactivity in people who are convalescent phase sera during the original SARS epidemic. So this sera was preserved, and this was found to be a really heavily targeted epitope. And in this paper, Wang et al., they reported that it was actually embedded in an immunodominant ADE epitope. So that's an antibody-dependent enhancing epitope. So when antibodies bound to this particular epitope, they actually increased a viral entry in their study. So it's a concern that somehow maybe this DG change is influencing an antibody-dependent enhancement, or this particular enhancement effect they felt maybe was due to when antibodies binding having a conformational change in spike. So it also might be related to that. So that's another hypothesis. And finally, it might be somehow contributing to resistance to immune responses to an initial strain in the current epidemic. So you might be infected with a Wuhan strain and then be exposed to something with this mutation, and maybe it's more readily allows reinfection. We don't have any evidence for that. We don't know if this is happening, but it's a possibility that I think needs to be formally considered until we figure out what's going on. So I'm going to just summarize on the next slide, slide 24, the case for positive selection of D614G. And I really think the most compelling thing is just the repetition of it. It's so many times in so many different geographic locales and at every level, city, state, country throughout the globe. A significant shift in the opposite direction was never observed, except in Iceland. And we think we understand what happened in Iceland because they did this interesting sampling thing where they really looked at travelers from high-risk European countries in early March. And so that would explain the pattern that we saw in Iceland. So if you have mixtures and you have shutdown and you have very well-established local epidemics where everything sampled is the original form, even if other events were coming in, like you might imagine in Washington State, people from Europe or New York were coming into Washington. These are happening in every county in Washington. They're happening after shutdown dates. And it's happening not only in Washington, but everywhere else in the world, including Hong Kong and Japan and South America. And you know, it's really the repetition that I find so compelling. And the fact that it's happening even after regional stay-at-home orders are in place. So this association with higher viral load in clinical samples is really intriguing. And not only have we found it with our Sheffield collaborators, but it was also observed in Washington when they looked at their data in the same way. And so we still need to figure out what possible mechanisms might be driving this global shift. And we don't know what those are, and we don't know if they're immunological, and I certainly hope they're not, because I think it would be wonderful if there's no impact on this, and the vaccines are blind to it, the vaccine responses, but we need to know this. It's really important to get it checked out. So if you go to the next slide, which is entitled Selection or Not, within 1 month, the dominant global form has changed. So you may not agree with me that selection is the best explanation for the data that I just showed you, but whether you do or not, it's undeniable that spike has changed and that the form that most of our reagents are based on was the original form, and now there is a single mutation that's quite an interesting one that has really swept the world. So I think it's really vital to understand whether this mutation does impact sensitivity to vaccine-elicited responses. Another thing that I think is really important to consider is that if this happened once, it could happen again. So it was seeded in places in early March when people were still moving around. Things have really shut down globally even at this point. But the fact that it can move this quickly just means that we have to stay on our toes. And the amazing people at GISAID and all the amazing people who contribute to GISAID will enable this. And I think this is really remarkable and wonderful. We can observe this, we can track it, we can see if it happens again, and we can stay ahead of the virus through this means. Another comment I wanted to make is that, people have said, well, we haven't seen this kind of thing before with the human epidemics that are related to beta coronaviruses. And the mutation rate is very slow. But I just wanted to point this out. The evolutionary playing field for SARS-CoV-2 is really vast, and it's a kind of a different world than we were in before. So the original SARS-CoV-1 epidemic infected about 8,000 people, and MERS, I think, less than about 3,000. But the May 14th WHO situation report estimated 4,248,389 SARS-CoV-2 cases. So this is now an enormous world for the virus to explore mutations in. So while there are only a handful of mutations arising in the course of any given infection in a particular person, across the whole global population, there's vast numbers of natural variants being explored and re-explored by the virus daily. So if there is something advantageous and it does start to move through the population, apparently it can move pretty quickly given what we've seen with this particular mutation and this particular haplotype. Finally, I wanted to talk just a little bit about recombination because I think it's an extremely important issue. So if you go to the slide called recombination, slide 26, it's a common natural evolutionary mechanism among coronaviruses, and it contributed to the ancestral evolution of the virus that was to become SARS-CoV-2 and pass into humans. And my colleagues, Fong Gao and Elena Georgi, and I have a paper in press talking about the emergence of this virus through recombination from past sarbecoviruses. We're good at looking at recombination. We've been doing it for years in HIV, and we had previously developed computational methods to identify recombination events in a low diversity setting of very, very early HIV and SHID infections. And so my colleagues, Tammy Bhattacharya and Elena Georgi, developed this program called RAPPER, which is able to identify recombination in this low diversity setting. And we use it to track recombination in vivo and sort of try to determine its relative importance to base mutation in the setting of HIV. And so we've been working to adapt our RAPPER tool to enable us to look for recombinant sequences among locally circulating forms in the regional epidemics of SARS-CoV-2. And this is still going to take a lot of work, but I think we've found very, very good candidates for recombination repeatedly. So I think it is a mechanism that is happening in the current pandemic. If you go to the next slide, there's an example of a recombination event in one of the Netherlands sequences that I'm showing to explain the kind of evidence we have for this. So if on this slide, you'll see at the top, I just put the whole SARS-CoV-2 genome. I just grabbed an image of that to emphasize the point that the red line and the blue line are two different full genome sequences from GISAID, and they cover the 30,000 bases of the full genome. And the one on the bottom, the sequence on the bottom, is what we're thinking is a recombinant child of these two parental strains. So the red just indicates the sequence that this person had, 415497, top one. The blue is that sequence. A tick mark comes in the bottom sequence when the red and blue differ. So any position where the red and blue are not the same, you get a tick mark in the child recombinant sequence. And so every tick is one of the positions where these two genomes differ from one another. And if it's a red tick, it matches the red strain. If it's a blue tick, it matches the blue strain. And you see it right around position 20,000. It goes from matching the red strain to going to matching the blue strain. There aren't more mutations in this recombinant strain than the ones you see. This is the only mutations it has. Mutations happen very, very slowly in these viruses. So the idea that you would serially select these handful of mutations, 7 whatever, it is just very hard to imagine in this background. In our wrapper statistic, we get a p-value of 0.0003 that this is a significant recombination event. This one happens to mix up our haplotype. So we go, remember I told you 3 positions, go with that G614G position. So the original Wuhan form is one of the strains. The new form is the other parental strain, and we've now mixed the haplotype. And so a better explanation for this mixed haplotype than spontaneously arising from mutation is recombination, because we're recombining these 2 forms. So this is one example of recombination, and in our preprint we put in 6 examples. There are a lot more. We just put in some to illustrate them. So if you go to the next slide, it says 6 examples of recombination. We had a couple from Washington State, and that very top one was kind of an interesting one because we could see a whole lineage coming off the putative recombinant. And then we picked 2 examples from the Netherlands and 2 from Iceland, and we did see many other examples of this kind of thing. We were just trying to indicate that we can find it, that they're highly significant, and they probably are recombinant sequences. So if you go to the next slide after that, recombination complicates interpretation of relationships in phylogenetic trees, and this is back to the first example I showed you. If you do the full tree, the recombinant sequence is that bright pink one in the middle, and the original Wuhan form is the blue, and the newer haplotype is the red on the bottom. And if you break the trees, if you break the sequences in half, so you do the first 20,000 bases on the middle tree and the last 10,000 bases on the right-hand tree, you see the recombinant form really moves around in the tree, and it goes with the recombinant parent quite precisely. There are more recombination events than this one in this data set and in many of the data sets we've looked at. So they are going to be confounding the trees to some extent. And I think it's important to consider this. So finally, implications of recombination on the last slide. Recombination events are present in the data, but we don't know if they're rare or common. However, because the 3 well-separated bases that distinguish the 2 haplotypes we've been discussing, the G clade, those are very rare. And since they're co-circulating so many populations, it suggests it isn't too common. But if it does occur naturally, it's indicative of distinct co-infections in a single host. And another thing to be kind of aware of is that recombination is a mechanism that viruses use to bring 2 favorable mutations that evolve separately in 2 different parents into one virus. So there's another thing to keep an eye on. It could potentially be an artifact of a laboratory contamination during PCR amplification. And the Salazar-Gonzalez paper that I cite there is one that came out of my friend Beatrice Hahn's laboratory, where we show that that can happen. And finally, whether it is a natural or sequencing artifact, they're definitely present. They can impact phylogenetic reconstructions, and we need to be thinking about them. It's just another thing to put in the back of our mind as we're interpreting trees and trying to understand how the virus is evolving. So finally, my last slide is acknowledgements. And although I've said this several times during the talk, I am astonished and amazed by the global effort that's represented at GISAID. And it's just a wonderful thing that so many people are working together to try to understand the evolution of this virus. And I've never seen a collaborative effort like this. So thank you to all of the people who collect the sequences and the clinicians who gather the data and the patients who give their samples, and GISAID for giving it back to the rest of the world. And then I'm going to talk quickly about my team here. Elena Georgi and Tammy Bhattacharya have worked on recombination. My husband, James Seiler, and Nick Kent-Gardner have done modeling and statistics. Nana Nanakaran is our protein modeling expert. Our little website that you guys are welcome to play with and look at was really pushed forward by Will Fisher, Hei-Jen Yoon, Werner Falter, and Brian Foley. The idea for a pipeline conception that has an immunology link, so we would find changes in spike that we would filter to the experimental community, was developed by David Montefiore and I, and Cecilia LaBranch has been helping us think about the immunology. And finally, I'm really grateful to the Sheffield team for providing clinical data linked to sequences and working with us on this and teaching us about how to think about possible sequencing errors. And we've had a couple of really lovely discussions that came from publishing in the archive first, and in particular, George Ellison and Barbara Imperiali have been really helpful and thoughtful. So I wanted to thank them and also thank our funders. Are you there?
Yes, Betty, thank you so much. We really appreciate your time in presenting this interesting data. I know we had a couple of technical issues. I'm not sure if you can restart your video at this time.
No, I can't.
Okay, I'm going to turn mine on, and we're
I
going to take
could, but I'd have to go to a different room and a different computer. It would take me 5 minutes. And I don't
Okay,
think
no
worth
problem. We're lucky to have you on the audio. We're just going to ask you a couple of questions, and then we'll wrap up. So we're a little bit over time. We received a couple of questions about whether these the G614 mutation, is it coming into the area? Are you hypothesizing that this is actually an introduction each time? Or rather, are these separate mutation events or a mix of both?
That's a great question. I think that it's definitely the same virus coming in because it's a haplotype. It isn't just one mutation. It's carried along with these other two mutations, and they're almost perfectly preserved. And my very first slide, I talked about that. So 98.6% of the viruses in the world either have one or the other haplotype with these three bases. And mutation is happening really slowly in these viruses. So the idea that you're getting all three of these recurring it is extremely unlikely to me. So it's really the haplotype that's moving into a population, I think, that's happening. So while mutation is happening really slowly, I do think this is selection because of what we've seen. And I think it's saying that selection doesn't have to happen slowly. Mutation slow, selection maybe not so much, is I guess my conclusion from this. And yeah, I do think it's getting separately introduced.
Interesting. Yes, that's related to another question that we got about selection pressure. Do you think that's associated with the lockdowns, and how rapidly would that occur?
No, I don't think it's associated with the lockdown, but there was a lot of discussion afterwards about people thinking, there's kind of a hypothesis that in Europe, there was a ski season, and the ski season ended in early March, and then people went home, like to Iceland, carrying the European form of the virus with them. And so the idea there is that you would have a seeding all over the world of people coming out of Europe and into different countries. And so the reason I thought the lockdown data was so interesting is that people seem to think you might get multiple reintroductions from the European. And so even if you had a really well-established epidemic, like in King County, Washington, maybe you get multiple introductions of this new thing and it would just shift. And the fact that the shifts were often occurring after the lockdown suggests to me that it wasn't travel-mediated multiple reintroductions, but rather a lurking presence of this form that had been getting transmitted in late February, early March, that started getting seeded in the population. But the transition from one to the other happened after the lockdowns when people weren't moving much. And that was also a recurrent pattern throughout the globe. So that's why I think the lockdown date was important, just because it shows that in many, many places, this pattern of transition from one to the other happened even after the lockdown.
Okay, and we've had a few questions about the applications to antibody therapies and for vaccines and for all of the therapies under development. Has the neutralization efficacy of convalescent serum been tested on these variants, and with the dependency upon the variant that infected the donor of the serum? Is there a relation there?
That's such a good question, and what I'm excited about, the reason I don't actually usually publish in archive, but I did this time, knowing this would be controversial, but I really wanted to get attention on this mutation, and the immunological community wasn't aware of it. So I think the epidemiology, the molecular epidemiology community was very aware of it, but my colleagues who were building the reagents to test the vaccines would call and say, you know, is spike changing? What do I need to look at in spike? And it seemed very clear to me that this was the one mutation that was coming up. So people have started making those reagents, but they take some weeks. You have to order them. Everything's slowed down with the shutdowns. And then you have to get the lab tests working and consistently working in the laboratory. And we have an amazing group of people working on this all over the world. And my colleague, David Montefiore, senior author on this paper, because he's one of them, and he wanted this pipeline to be aware of any mutation in spike that should be checked. So they're working on it, but they haven't gotten there yet, and hopefully that will happen within a month or two, maybe even sooner. So what I know is in progress right now is they're trying to make the viruses, they're trying to make pseudoviruses, they're trying to make the protein, they're trying to crystallize the protein in both forms and see if there's a difference. And we don't know if there will be. We're hoping that there won't be. And so then these reagents will be available as these first test results are coming out from the first promising vaccines. So pretty much, to my knowledge, all of the vaccines are made in the original Wuhan form. And it will just be a great reassurance to know that these don't make an impact if they don't. If this mutation does make an impact, then we can deal with it. Then we can start thinking about, okay, do we need to shift the vaccine? Do we need to modify it in some way to accommodate this? So either outcome, I think we need to know. And people have started working on this in March. It's just taking a few months to get everything going. If this was a virus that everybody was familiar with, it wouldn't take so long.
Sure.
You know, if this was HIV variant, we could have had it within probably a few weeks for testing, but there's just a lot to be worked out. Once the system is worked out, other variants will be easier to look at if they arise and are of concern. There also, you know, might be variants of local concern, and we're trying to, that's part of our pipeline effort. So we not only look at the global, but we look to see, okay, is there a variant arising in a community, like there was one in the signal peptide that was showing up in Hong Kong, just in Hong Kong, nowhere else, but it seemed to be increasing in frequency in Hong Kong. So, you know, keep an eye on that one. Does it spread out of Hong Kong? Does it really take over in Hong Kong? And so those kinds of things we just want to monitor, not to be alarming, but just to know what's happening and have things that rise to a level of prominence or local prominence, that our variants be available to make sure the reagents work in those places. So the question for vaccines and the question for antibodies is really the same. You know, we'll have the antibodies isolated from convalescent sera and from vaccines in primates, and we can test them against both, but that'll probably be a month or two before we're really able to get there.
Sure. Okay, so one last question. We've received a couple of different versions of this question as to whether these mutations, does this make the virus more virulent, more transmissible and infectious, and what are the next steps for your work?
It's a great question. So I think it makes it more transmissible. I think that the data speak for themselves and are very compelling. And I get more compelling every week when I reevaluate the GISA data. But so one thing is just to keep an eye on spike, and then I'm working with several colleagues on trying to see if they can get an experimental difference. So I think something about this is more transmissible, and I gave you guys a couple hypotheses, and I have no clue which is the right hypothesis. So it might be an immunological effect. It might be something about the virus becoming slightly more infectious, and maybe even just a slight advantage could cause a shift in the population. I really don't know. So one of the things for us will be helping with the statistics and analysis of the 614 variant. Another thing will be over the next year or two, continuing to monitor changes. Another thing will be to expand outside of spike and to go into other protein regions. This particular haplotype also has a mutation in the polymerase, so it might not even be spike at all. It might be the polymerase that somehow modified and making the virus better able to succeed. I'm not sure. So that's also going to be tested. Ralph Eric is going to look at that to see if he can see any impact of that mutation on the way the virus grows in vitro. So I think right now we're just at a point where with this particular mutation, we can say, please explore these things experimentally and clinically. I guess the clinical data from Sheffield and Washington I found greatly comforting. In fact, I was finally able to sleep again when I saw it because there was not any overt association with hospitalization. So it did not seem to be associated with disease severity, at least at the level we could look at with 1,000 patients now. So that's greatly comforting. But it does seem to be associated with higher viral load. I guess that would point to a virological of this mutation.
It's so interesting, yeah, to see how all this data come together. Okay, well, thank you, Dr. Korber, so much for sharing your insight and knowledge on COVID-19 with us today. And I want to thank all the attendees for your participation. We realize everyone is especially busy and you have many obligations right now, so we really appreciate your taking the time to join us for these discussions.
Thank you. Thanks for letting me talk about this.
Thank you so much. I invite all of you to join us in 2 weeks from today on our next Global COVID Lab meeting, which is scheduled on June 4th at 10 a.m. Eastern Daylight Time. Our next speaker is Dr. David Weiner of the Wistar Institute, who will talk about the recent progress of synthetic DNA vaccine candidates for SARS-CoV-2. And if you're interested in more research on COVID-19, please sign up for our HVP COVID report. In this newsletter, we present insights from experts all around the globe and highlight the latest scientific articles and data. And lastly, please feel free to visit our website and follow us on LinkedIn. We'll upload a recording of this webinar there as well once it's available. We have a great day, and we hope that you'll join us again next time on the Global COVID Lab meeting. Have a great day, everybody. Thanks.